a lot of studies in Alzheimer's disease and other dementia, we use mouse models, of course, and other animal models. And the thing is, mice, they do not naturally develop Alzheimer's disease, for example. So what we have to do with mice is actually we have to, introduce a lot of different disease mutations. Sometimes you have to introduce 5 mutations in order to see a pretty severe phenotype. So it's a little bit artificial, but granted, that's the limitation that we have with the disease models with mice. So that's what we have to do. So in terms of the human stem cell model, of course, it's human. So is in terms of the species is more relevant. What we can do as compared to animal models is that we can derive the human stem cells from a human patient who's still alive. And that human patient might or might not have any disease mutations. And what we can do is we can model what's happening to the patients in a patient-specific way. So let's say the patient has a certain AD mutation, and then you take some blood or skin cells, and then you can reprogram them into stem cells in vitro. And those cells also will carry the same mutation. And you can start study what's happening in the dish. Now, of course, it's not a replacement of animal models. The human stem cell model is meant to be a complement of animal models. There are also many caveats about human stem cell models. For example, it's in vitro, it's not in vivo, so it's very often a monoculture. So it's not always very relevant, but it has its advantages and disadvantages.